Hi everyone, welcome to the new series looking at structural variant analysis. Today we're going to be performing matched tumor-normal structural variant analysis using this tool called SvABA. And I'm going to be walking you through the entire process from installing all the necessary packages as well as interpreting the outputted data from SvABA to plot in a Circos plot, as you can see here, which shows the various structural variants that are present in a genome. So taking a step back, for those of us who are not familiar with what a structural variant is, at least in the context of the human genome, a structural variant is a large genomic change, typically greater than around 50 base pairs, although it doesn't have to be, it can be smaller. So there are 5 main categories of structural variants. First is what we call a deletion, or depending how big it is, can be a deep deletion. And this is really when a large chunk of a chromosome is essentially just removed. And this can be megabases or just a couple thousand base pairs, but generally we're just seeing a loss of DNA sequence in 1 or 2 of the chromosomes for a particular chromosome. And so the second one is looking at insertions. So this is when a fragment of DNA is erroneously inserted into the middle of a chromosome, as the name suggests. So not a super confusing structural variant here. The third is called a translocation, and this is when 2 chromosomes, say if you have chromosome 1 and chromosome 2, and they swap arms. That is a translocation, particularly a balanced translocation if there's no information actually lost, they just moved it. So essentially translocation is when chromosomes swap arms. The fourth type of structural variant is called an inversion, and this really is when you take a particular region of a chromosome and it is reversed. And so, as you can imagine, if you reverse a particular fragment of segment of DNA, this is going to affect the order in which genes are read and affect gene function. So these can be deleterious as well. And lastly, we have these duplications, which is pretty common for oncogenes like MYC. You can see that particular oncogenes will become duplicated over time within a given particular chromosome. This is also found in diseases like Huntington's, when a particular repeat region is duplicated over and over and over. And so this is basically just when a particular region is copy and pasted within the same chromosome. And so why do structural variants matter? At least for me, in the context of cancer, this can disrupt genes, a regulation. It can remove a particular protein from a cell. It can also amplify up a particular protein. So this can basically toggle gene expression quite dramatically. And not just cancer, but this can be important in multiple different diseases. Like I said, for instance, Huntington's duplication is very common. And also, these can be challenging to detect with standard next-generation sequencing, because next-generation sequencing, these fragments are relatively small. So it can be a little tricky to kind of map these large deletions, or just trying to characterize these large structural changes, particularly in regions with a lot of repeat DNA sequences. And so, just high level, this is the landscape for structural variants and what we'll be trying to detect in a matched tumor-normal sample. And so the tool that we'll be using today is called SvABA. This stands for Structural Variant Analysis by Assembly. This is a tool developed at the Broad Institute. It claims to have higher resolution than alignment-based SV callers. I believe it. So the difference between an alignment-based SV caller and an assembly-based SV caller is that alignment-based SV callers are going to take just the reads and compare those to the reference. And if they see any anomalies with the alignment of those reads, for instance, one read is aligning to chromosome 1, another read is aligning to chromosome 10, it's going to be confused, saying why are these two paired reads aligning to two different chromosomes, and depending on how many are actually doing that, it will call that a translocation. The difference is with an assembly-based structural variant calling tool, it's actually going to take the fragments and outside of the reference, it's going to create these longer contigs, which are many reads stitched together. So it's essentially assembling its own genome based in regions that have anomalous read alignment. And then it's going to take these larger contigs and compare them to the reference. And so this just increases the ability to detect complex structural variants and can recognize medium and large indels. What this does is just takes short read data, and it has a couple different analyses. It can do tumor-normal analysis like we'll be doing today, and it can also do germline analysis. I've never actually done the germline analysis, so if this is an area that you all are interested in learning, I can try to go take a look and do it. But just for today, I'll stick with what I know, and we'll be doing tumor-normal structural variant analysis. And just generally, these assembly-based tools require more compute than alignment-based tools, so just keep that in mind. And so why would you use SvABA over other tools? Mostly just because it's an assembly-based tool, so it has a higher sensitivity for complex rearrangements, particularly translocations and deep deletions. These can be pretty tricky to call if they're in a region with a lot of repeats. So performing this assembly just makes calling these a little bit more confident. And it's a tool that's ideal for cancer genomics. And I guess last point that I've read about is that the breakpoint resolution, meaning the exact nucleotide that a breakpoint occurs, is actually much clearer using these assembly-based tools. And so again, just trying to do a little compare and contrast about why you should be using SvABA, or maybe in what particular situations would you want to be using SvABA versus another tool. So assembly helps resolve ambiguous regions such as repetitive sequences or overlapping rearrangements, which can be problematic for alignment-based approaches that rely heavily on correct initial mapping. But assembly-based methods require more compute, and they tend to be slower, and they're not always ideal for high throughput pipelines. So SvABA tends to be better for specific research applications. Say you have a particular sample that's giving you a headache and you don't understand what structural variant's driving a particular phenotype. SvABA is great for that particular case. However, if you have a ton of different samples, say you're analyzing a patient cohort of 200 matched tumor-normal samples, SvABA may not be the best approach. You may want to use a quicker and less compute-intensive tool like Dragon or Manta or Deli to perform these kind of clinical large cohort structural variant scans. I guess it really is up to the user. In this case, we do a lot of like low sample number research-based structural variant analysis, so we focus on SvABA. But I've also used Dragon too, and Dragon can be a lot quicker than SvABA too. So just keep that in mind. There's pros and cons to both, but I just wanted to highlight the differences between these two, and hopefully leave it in the comments if you have any more questions. Happy to point you to a particular tool. And so then just generally running over what we will be doing today. First, we're going to do a lot of the upfront structural variant calling in an AWS instance. So we're going to launch an AWS instance, and then we're going to have to install a ton of software. From a super high level though, we're just installing SvABA, samtools, and BWA. There's just a lot of extra junk you gotta install onto the instance for that to install properly. Next, we're going to actually run SvABA once we get everything all set up. We'll talk about what input files you need, what reference FASTA you'll need, and what the output files look like. And then once we actually run SvABA, we will plot the data using a Circos plot tool called Circos. And this will be done in R. So we'll actually take the data out of the AWS instance, download it to our local computer, and do all this analysis in R. So yeah, with that, we'll get into the analysis. Okay, so now we're going to create an AWS Linux instance and install SvABA onto it so we can perform the tumor-normal structural variant analysis. So the first thing we need to do is go to our AWS portal and then navigate to our EC2 so we can launch an instance. We'll come here and then we'll just click launch instance. I'll give it a name, SvABA instance. Like I said, it's Amazon Linux. Since SvABA is relatively compute intensive, I like to use the T2X large with 16 gigabytes of memory. I've found this is sufficient to perform the analysis. And you also need the key pair so you can actually log into the instance, and you also need an existing security group, the one we've already made for the Emory security network. Also, we have to change the storage. Since whole genome sequences are around 200 gigabytes, and we're going to be comparing tumor-normal, we need a little bit more than 400, and I like to be on the safe side, so I make it 800. And we also need this EC2 to S3 access just so we can move files between EC2 and S3. But other than that, oh wait, nope, that's everything we need to do. Everything else stays the same. So we'll let that launch. Come back here. We see our SvABA instance is loading, and now it's running. So I'll come here and get this connect SSH command, and we'll open our terminal, open a new window. I'll just use this one. I'm already in the folder with my PEM keys. So here are all the PEM keys that I've downloaded. I have lots of different ones, but the one I'm going to be using today is the G. Cooper Hong Lab one. So I'll come back, copy this SSH command. Sometimes I get these funky little squiggle 00s on either end, and I don't know why. It's very random. But we don't need those. So I'll delete those. And then we'll delete this IP address, which is not correct. We actually have to use the private IP address to log into this instance. And again, I get these weird squiggles that I need to delete. This probably shouldn't happen. This is a weird thing that happens on my computer. And so we get this remote host identification has changed. This is just that SSH keygen command. I do this almost every time I log in. So I don't really know why I do it, but I do it. And now it should work. All right, so we are in our AWS Linux instance now. We're no longer on our local computer. But before we install SvABA to perform the structural variant analysis, we first need to install a few dependencies, and we're going to start with htslib. This is a C library that's used for reading and writing high throughput sequencing data, and it's used in a lot of different packages that do sequencing analysis. So starting off though, just so we can actually pull the repository onto our instance, we'll need to install git. And so we'll just use sudo yum. This should be relatively quick, in theory. All right, so now we have Git installed. You can see it's in our path now. You can see Git. You can now pull any GitHub repository onto your instance. Next, we're going to install some basic development tools that's needed to compile software from its source. Generally, what compiling is you're just converting human readable code to binary so you can actually execute it. So that's what we're downloading here. So we're going to do group install, development tools. Why? And so yeah, this is just going to let us compile packages so we can actually execute them. And then next, we're going to install some compression and networking libraries that HTSLib depends on. Yeah, HTSLib needs a lot of different packages, you'll see. That needs to be installed for HTSLib to work correctly. But this is a really common thing we need to install, so this is not used just for SvABA. So save this video. If you're ever needing HTSLib again, you can install it the same way I'm doing it right now. Okay, so we have the development tools installed. Now we need to install this compression and networking libraries. So, devel, EC2 devel, xz devel, and curl devel. That one should be a little quicker. Okay, so now that we have all these kind of tools already installed in the instance, now we can actually pull the HTSLib repo onto our instance and install it. So we'll use the git command that we downloaded earlier, and we're just going to clone the HTSLib repo onto our instance. And this is just the link to the HTSLib GitHub page. You can get this from the internet if you type in HTSLib. And so this is just going to pull all the human readable files that are on the GitHub repository. And now we're going to navigate into the repository. And there's also some additional external code dependencies that HTSLib uses, so we're just going to make sure those are also downloaded as well. Oh, I have an S. Don't need the S. No. There we go. All right, so now we have all the external code dependencies installed. Now we need to compile the HTSLib repository. So yeah, this is just taking all of the human readable code that the developer wrote and converting it to binary so we can actually execute everything in there. This is a pretty common technique used when you download repos, especially with C languages. You pretty much have to compile anything if you want to actually execute it. I will say compiling can take a little bit of time, so I might jump forward in this video and wait for it to finish. Okay, so we have officially compiled the htslib repo, and now that it is compiled, we're going to install it. So that's pretty quick. Now we have htslib installed. We now need to install CMake. SvABA uses CMake to configure its build system. So this is also a relatively common package you'll need to compile a lot of bioinformatics packages. Okay, so we have CMake installed, we have htslib installed, we have Git installed, as well as all the extra developer tools and stuff like that. So we have everything installed except SvABA. So next we will clone the, let me navigate out of htslib first. So we're going to clone with the git command the SvABA repository. I believe this is version 1.2. Okay, so now you can see the SvABA repo is in here. This is everything that's on the GitHub page. So now we need to create a build directory and navigate to that. Oh, make the build directory, it's not already existing. So we navigate to the build directory, and then we have to actually compile SvABA, we have to point it to where htslib is. So the way you can determine where htslib is located, typically htslib, when you install it, is in this user local, but I'm just going to confirm that by trying to find all the file names with hts in it. Okay, and so this is just confirming htslib is installed in this local folder, and these are the subsequent files or folders that were downloaded. So now we're going to use the CMake command to compile the SvABA htslib. And so here we're just pointing it to the directory for htslib, which is required for reading and writing high throughput sequencing data. Okay, so everything has been put into this build folder, and now we just need to compile it again. So this is going to take a little bit of time, so I'm going to let this run, and I will see you in a bit. Okay, so we have successfully compiled the SvABA build repo we made with CMake, and we can run SvABA now, but just so it's easier to call from other directories, I like to add it to the path, so I can just call it up by typing in SvABA. So that's what I'm doing here. I'm just pointing this directory to the path. So we can type in SvABA and run the command from anywhere on this instance. Okay, so now if we type in SvABA, you can see it's installed, and if you type in help, these are all of the help commands it brings up. So we finally installed SvABA, so next we're going to actually pull in our whole genome sequence, BAM tumor normals, and perform structural variant analysis. So now that we have SvABA installed on this instance, we need to pull our whole genome sequences on here. So I have some preloaded whole genome tumor normal data on my S3 bucket. I downloaded this from Genomic Data Commons, and I'll likely upload a video showing how to download that, but it'll take a while to download, upload to AWS S3, and then pull into here. So I'm just going to show you how to move it from your S3 onto the instance. And also just to note, you do need to have the index as well as the BAM file. So I am going to pull these onto the instance. And so I'm going to use the nohup command just so that this runs in the background. This will take a long time and my instance may time out. So I'm just gonna use nohub to make sure it keeps running even if this times out while it's running. And so we'll copy that BAM file. Oh wait, I'm gonna make a new folder so it's not in the build. So I'll come here, I'll make directory SvAnalysis. Okay, so we'll put the BAM files in this SvAnalysis folder. So AWS3, copy, make sure I put the nohub. I'm going to copy this folder or this file and put it on this instance. And I'm also going to add this and sign just to make sure I can keep running commands while this download occurs. Okay, so that'll start that download. And then we will do the same thing. So this is the normal here, indicated by the end. We need the tumor as well. So, also just to note, these samples are rhabdoid tumor normal pairs. So we're expecting actually to be few structural variants outside of SmRCB1 variation. So rhabdoids defined by biallelic SmRCB1 dysfunction, and so we're hoping to identify some sort of structural variant at SmRCB1 in both alleles in the tumor sample here. Oh, it looks like I actually copied the normal. Let me copy that. So I'm actually going to kill this job. Let's see, is it downloading twice? Yeah, I just want it once. So I'm going to kill this job. Okay, so that's only one now. Should be downloading. And so then we're going to download the tumor now. S3 copy. Yes. Okay, that's the tumor. We'll make sure which one of these is actually still downloading. Okay, we can delete this one. This is not the one. This is the one I terminated. So remove that. Just make sure these are both downloading. Yeah, you can see they're growing in size. So they are currently downloading. We also need to bring over the index files. So AWS S3 copy. These are much smaller. I need to put the period to indicate we want it in this folder, as well as this one. S3 copy. Oh, did the same thing. I copy this again. AWS S3 copy. I moved the tumor index twice, so I'm doing the normal again. Okay, so we have both of the indexes for the whole genome alignment files. These are still downloading, so I'm gonna let those do their thing, and I will start back up when those are finished, moving from the S3 onto this instance. So now that we have SvABA installed on our AWS instance, we need to go find the specific reference that the tumor-normal aligned BAM files were actually aligned to. So I had actually tried doing structural variant analysis using the GRCH38P13 reference that we had used previously in the Dragon RNA-seq quantification and alignment video, but when I ran SvABA, it actually kicked back some errors showing mismatching names between different contigs within the reference. And so this is me still making a mistake on this, but I just wanted to re-emphasize when doing any sort of sequencing analysis, the reference file and version has to be the exact same throughout every single step of the analysis process. So I went back and found, okay, which reference file did GDC use to align these tumor-normal whole genome sequences? And we found that they were using the specific GRCH38 reference version. And so we're going to have to download this file, move it onto the S3, and then move that onto our AWS instance, and then we're going to have to do some indexing. But first, we are going to download this file. It can be a little glitchy, but that file is now downloading, and when that's finished, I will move that onto my AWS instance and do some indexing. So I'll see you when that finishes. Okay, so that's reference finished downloading. So now we're going to navigate to our AWS console. We'll come in to our S3 buckets, and we will move this FASTA file onto our S3 bucket so we can move it onto our AWS instance. So I'm going to make a new folder. Just so anyone in my lab who needs this. Oh, I had done this previously, but I'm going to do it again just for you all. And then I'll just upload that folder, that file we downloaded right here. It'll upload. And so this will take a little bit of time, so I will see you when this finishes. Okay, so that file we downloaded from the website is now on our S3 in the folder I just made. And so now we're going to move this onto the EC2 instance. So we'll do AWS3 copy here. There are some spaces in this file that we downloaded, so we just have to put apostrophes around it so it knows what's actually the file here. And since we downloaded the tart and gzipped file, we're going to need to basically undo that. And so we'll SvABA. Alright, and this should untar and unzip this particular reference FASTA. Okay, cool. So now we have our untarred reference FASTA that GDC used. So I'm just going to remove the original one. And so now we have our reference FASTA on our AWS instance. Next, we will need to index this particular FASTA file using BWA and samtools. So I'm going to walk you through how to install BWA, how to install samtools, and this is all going to be done using bioconda. So we'll get started with that right now. Okay, so next order of business is indexing this FASTA file we downloaded using BWA and samtools. And so in order to install BWA and samtools, we need to first install miniconda, which is a repository used to install lots of other bioinformatics packages. But so first we're just gonna load on miniconda onto this instance, and then we're gonna install it, configure it, and then we'll use that to install BWA and samtools. So the first thing we need to do is pull on the repo from Anaconda's website. This is a pretty common package manager that bioinformaticians use. There's tons of different packages that are included on miniconda that are not just for this particular analysis. Pretty much whenever I launch up a AWS instance, I install this miniconda repo, just so I can download whatever I want when I want. And so this will just download all this associated files. I'm actually going to move this up one level just so I don't mess with all this stuff we have in this folder. Okay, so we have it up here in this kind of home directory. And so now that we have downloaded this repo, we need to run it with the bash. And so this is essentially just installing it. And so there's going to be a lot of different prompts. Pretty much just hit yes whenever it asks. Okay, so then confirm this location. Yes, this looks fine. Let it do its thing. And so this is going to set up the shell so that it's actually able to recognize conda. Do I wish to update the shell profile? And yes, I do want you to update the shell profile. Okay, and so now miniconda 3 is installed. And before it will work, you actually have to close and reopen the particular shell. So we're going to do that. You see me recording, and you can see my horrible tabs. But so we have to basically relog back into our instance. Let me navigate back quickly to where I keep my PEM keys and hope that this SSH command is right. I think it is. Yes, it is. Okay. And so now that we've relaunched our shell, conda is now in our path. So that is good. That means we've installed miniconda. And so next what we're going to need to do is do some configuration steps. I found that there was just some errors when I did not run these, so I've always just kind of ran these config steps. So it may or may not be necessary, but a long time ago I found that this fixed the problem, so I always do it now. Leave it in the comments if you think it's not necessary, but I find that it helps. And then conda forge. Okay. And so now that we've configured the conda installation, we can now install BWA using the sub repo called bioconda. So this is going to be a lot easier than installing, for instance, SvABA. These are really going to be one-liners, and you'll have access to a plethora of various bioinformatics tools, such as aligners like BWA, or just go look at Bioconda's website. You can see all the crazy packages they have just ready to install. So just say yes to all this. And now BWA is in our path. You see how easy that was? And so next we're going to have to install samtools as well, so we can index with that. Okay, that should be almost done. Alright, and we just make sure samtools is indeed in our path. So now we've installed samtools and BWA. So next we're going to have to index the FASTA file that we just downloaded. And so the commands that we'll be using, first we'll do samtools since it's quicker. FAIDX. We just select our FASTA file. And let it do its thing. This takes a little bit of time. but not as long as the BWA. BWA takes a while to run. And so I'm about to run that. So instead of FAIDX, we're just going to say index here. So I'm going to run this command and let it do its thing in the background with the no-hup and ampersand, and I will see you when this finishes indexing. Okay, so BWA finished its indexing for the GDC human reference genome. This took about an hour or so, a little bit over an hour. So because of this, my shell session timed out. So we're gonna have to reload the SvABA build directory into my path, just so we can call on the SvABA command from anywhere in the instance. We'll do build. That is the path, I believe. Let's see if we have. Okay, so SvABA is now in our path, so we can directly call it. And so now that we have our reference, we can just run SvABA. The command is relatively simple. Just call SvABA, you want run. And then we provide, oh, let me pull up the, yeah, here are the file names so I can copy and paste. I like to copy and paste file names just so I don't screw anything up with the typing. But here we just do T and provide the tumor file. Here we provide, for the N option, the normal whole genome file. And then we provide G for the genome. And then we just give it the FASTA, and it will automatically recognize all the associated indexed files. And then for the A, you just put what the prefix you want your output to be in. And so, since this takes a while, I am going to run this in the background again with this no-hup. I'm going to copy this command and delete my current no-hup file, just so we don't have any overlap. So I'll remove this no-hup. And now we'll run this again. So all the output will, all the verbose output will be put into this no-hup file. And so SvABA is running. This will take a little bit of time, so I'll let this finish and I will see you when it's done. Hi, SvABA officially finished. It actually ended up taking 36 hours to completely finish. So just keep that in mind when you are running this. It's going to be around 36 hours per whole genome, human genome, whole genome sequence at about 30x. So takes a long time, hence why this is not very useful for high throughput clinical data. But when you are looking at the output from SvABA, you see a lot of these different files. Basically anything with SvABA output is the output of the run. Really, what we're going to be interested in here only is this SvABA output somatic sv.vcf. And this is going to be looking at somatic variants that are present in the tumor. We're not really worried about germline because we're not doing that analysis. So we're just going to quickly move that over to our S3 bucket. And so we'll copy that. However I moved it, but I'll just show you how I'm doing this. So we move that on over here, and we are going to download this onto our local computer, and we're going to process it, ideally plotting it with a Circos plot, and kind of talk through the data a little bit. So we'll download that. I'll show you, whoop, we'll put that away. And so then the way I do this is I will then just copy this into the SvABA tutorial. And so I'm not going to replace this since I already have done it, but that's going to be how you move the data from the S3 onto your local computer. And so then we're going to now move into R. So I actually have already set this up. And so here's the code I use to process these SvABA structural variant data and use it to process into a Circos plot. But first, I just wanted to kind of walk through the data a little bit. So I'm going to read in this SvABA VCF file. And just to note, if you're not familiar, if you just want to read in a file like this, you have to save your R Markdown file in the same folder as your input data. So since this SvABA sv.vcf is in the same folder that I'm saving this R Markdown file, I can just read it in without putting the absolute or relative path. So just a quick tip, if you're doing R analysis, I like to keep my R Markdown in the same folder as all the data I'm analyzing. It just makes the whole code chunk easier. So if we look at the actual output of SvABA, if you're not familiar with a VCF file, they're kind of organized by chromosome and position, and there's going to be a lot of additional information for each of these structural variants. Each line here represents one structural variant that SvABA has identified, and yeah, so I'm going to focus on one of these, in particular this one in row 19, as this is the one that affects SMARCB1, and since this is a rhabdoid tumor sample, which is known to have SMARCB1 alterations in it, I'm going to focus on this one and kind of walk you through how I interpret this data and how you can apply this to understand which genes are actually being affected. So that's what we're going to do next. Hey, so I decided to walk through how I interpret one particular structural variant identified by SvABA. When you run SvABA, you're likely going to get a lot of different structural variants. It's not just going to be one like this, but I'm walking through one just so we can get an idea of how to interpret these. So on the top bar here, I just copy and pasted the first 5 columns of the output from SvABA in that VCF file that we read into R. In the middle here, I've shown a graphical representation of chromosome 22. This is the chromosome with SMARCB1 on it, and denoted where the break-ins are occurring based on this particular structural variant that's identified. And on the bottom here, I'm just showing the structural variant mate. So each structural variant is going to have 2 different rows in your SvABA output, because if there's a break end, you're going to have abnormal realignment on both ends of that break end. So you're going to have 2 rows for every break end structural variant that SvABA identifies. And so now, jumping to the top here, walking through each of these different columns. So on the left, pretty self-explanatory. This is the chromosome it's identified on. This position here is showing the beginning of the break end event that SvABA identified. This ID column here, not important. This is something SvABA applies to each structural variant. And then this reference column here is just showing what nucleotide the structural variant begins at. And so really the important bit here is in this alt column. This is showing where the read alignment was abnormal. So SvABA identifies these by seeing which reads are mapping to 2 different regions of the genome. So when you're aligning a particular fragment of DNA, you're expecting it to just match one region of reference, because that's how typically alignment goes. But if you align a read that should be a contiguous sequence of DNA, and it's mapping to 2 different regions, in this case, half the read aligned to this part of the chromosome, and the other half is aligning to this part of the chromosome. So obviously something weird is going on here. And so that's really what this line is showing, is that some read was aligned to this part, and then at this particular nucleotide, it suddenly started aligning to this particular upstream region around 2 million base pairs upstream of the beginning of the read. And so kind of walking through what these brackets mean, this is just showing that at this G nucleotide, this bracket is showing where the break-in is directionally going. So where the the little pointy bits are pointing is showing where that break end is pointing towards. And so this G is suddenly aligning to this particular region here. And so that's kind of what the directionality of that breakpoint is. And then similarly in this mate, on the bottom here, you can see how the position here in the first part of the mate is matching to this number here, and vice versa. The alternate allele here is now the position here. And so really this is just showing that this was one, so this row here is showing that this break end event is occurring, and then by nature you're going to have two break ends if you're having some weird event occurring. And so this other line is just showing the break end event that is occurring here. And so the reason these brackets are pointing this way is because this break end is pointed in this direction. And so the reason why that we know this particular structural variant is affecting SvABA1 is because the break end is occurring on either end of SvABA1. So we know that either this particular region of DNA, it could be either inverted, meaning that the orientation flipped, or it could be that this entire region was completely deleted. And so my gut is thinking that this is a deletion because SvABA1 protein we know is gone based on the diagnosis. This is a rhabdoid tumor, we downloaded it as public data. So SvABA actually won't tell you this. You have to do some downstream analysis, looking particularly at coverage in this particular region. But yeah, so just take a note, SvABA only outputs break end. It will not specify if this is an inversion, translocation, yada yada. You kind of have to do that digging on your own. So that is pretty much everything I wanted to cover. I guess one other thing, I'll show this, actually I'll show this right here. But first of all, this is that break end notation I was talking about. You see how every single structural variant here is denoted by break end. This is specific to SvABA. Another tool called Dragon actually will classify these by different types of structural variants, but SvABA will not do that. Another thing that I always keep an eye out for is this particular 1/1 or 0/1. This is just letting you know, is this happening on both chromosomes? And so if it's a 1/1, that is happening on both chromosomes. If it is 0/1, that just means it's happening on one chromosome, or heterozygous or homozygous is the terminology we use in genetics. So yeah, that's pretty much everything you really need to know about the output to interpret it. And so next we're going to dig in to how we can plot this output in a Circos plot, which is a really common way to represent structural variation, particularly in cancer genomes. All right, so we'll get to that. Okay, so here I'm showing the end goal of what we're trying to plot here. What this is called is a Circos plot. It's like circus with an O at the end. What this is showing is every chromosome is in this circular shape, and the length of the chromosome is denoted by this line. As you can see, the chromosome 1 is longest, and they get progressively shorter as you go along. If you're a human geneticist, you know how this is formatted. And what this is just trying to show is for each of these lines that comes up here is denoting a break end event. So this is essentially kind of a very, very skinny kind of arch that is arching between the beginning of the breakpoint, goes up and connects back down to the other end of the breakpoint. In this particular sample, there actually aren't any balanced translocations, but if there is a balanced translocation, meaning the chromosomes will swap entire arms, you'll see that the Circos plot will create this longer arc. Say if there's a translocation between 12 and 22, you're going to see this long arc connecting these two. We don't have that in this sample, but this code will plot samples that do have that all the same. So I also, up here in the corner, I have a legend for deletion, insertion, break-in. SvABA output does not denote if it's a deletion, insertion. It says everything is a break-in. So This is more set up for other tools that will actually annotate the structural variants for you, but we really don't need the legend here, but it's there. This is all set up just for so you can use multiple different structural variant tools. So here I'm jumping into the actual code. I'm assuming you have some baseline knowledge of how to install packages, load them in, and generally use R in general. I might make a video of just a crash course in R in the future, but if you have any questions about this, please leave them in the comments. Happy to help you get this up and running. But here in the top are the packages that you're going to need to perform this analysis and plotting of the structural variant output. And so yeah, I'm not going to type it out like in previous videos. It's a lot. I'm just going to kind of walk through each line so you conceptually can understand what I'm doing and not just hitting run. So if you do need to tweak the code yourself, hopefully you can get this figured out for your own data set. Okay, so from the top here, I'm just reading in the SvABA VCF output, and I'm commenting out anything with a hash. The VCF files have a very long header that we do not need for this particular plotting, and I'm just reading it using this TSV function since it's tab separated into this data frame called VCF_DF. Next, I'm just doing a quick filter for all the structural variants that passed SvABA's kind of statistical threshold for what is a real structural variant. Luckily, they actually already do this. Other tools don't already do this, so if you're using another tool, I always like to just filter for ones that pass. You don't want ones that don't. And so then I'll pull up this VCF filtered just so you can see kind of what I'm doing under the hood. And so what I'm doing in these two lines is just extracting the first two columns. So I'm just pulling out the chromosome numbers here, as well as the position for the beginning of that break end. And so pretty simple lines here, that's all I'm doing. And I'm putting that into like a list of values, not a data frame. And then since the alternate column, I'll show you what that looks like. This alternate column is a little bit trickier in that you can't just explicitly extract it. It has this T, the brackets, chromosome, colon, but we only really want this and this. So we're gonna have to do a little finicking with these particular, with this column to extract those two values and leave the brackets and the reference allele outside of it. And so I'm just extracting that column so we can do some downstream filtering. Here I'm just making a data frame so it's easier to loop through. And then here, okay, so we're basically going to take this alt info df. So I basically just extracted all of this and we're trying to pull out chromosome and position. That's all we want. But we have to create first and we have to initialize a numeric vector for the breakpoint positions and the chromosomes. So here I'm just kind of setting up a vector to store the positions of the breakpoints. And here I'm just reading in the same chromosomes of the beginning of the breakpoint. It may change on the line, but I believe in this particular sample, they're all structural variants within one chromosome. We're not seeing any cross-chromosome structural variants, which can happen, but not here. This will work even if you do have cross-chromosomal structural variants, though. And so then, while this code chunk may look a little scary, it's really quite straightforward. I've set this up in a way that is able to handle particularly Dragon output in that the alternate allele value looks a little different depending if it's a deletion, insertion, or break end. So I'm just kind of doing this for loop where I iterate through every line in this data frame called alt info df, and essentially using regular expressions to pull out only the chromosome and the position. And so I won't get super into the weeds here, just trust me that this will work on this particular data frame. But if you have any questions on this code and it's not working for you, please, like I said, leave something in the comments and I'm happy to help you get this working. So this is essentially, so we're taking these two kind of empty placeholder vectors, and this function is just reading in the values that we want into those placeholders. That's all that's going on here. And then here we are just creating a data frame of all the different breakpoint positions and chromosomes and just to one data frame so we can process that downstream. And I'll actually show you what this looks like at this point. Let's let that run. So now we're just looking at a 4-row data frame, and that's really all we're going to be plotting. And so then an issue with Circolize is that everything has to be in chronological order. You can't have the chromosomes out of order, and if you have two breakpoints on a chromosome, the one that comes first has to be the lower position number. And so what this code is really doing is just ordering everything so that if the first position is greater than a downstream position, it's going to swap those chromosomes or position pairs. And this is just standardizing the ordering for consistent plotting. This is kind of just a quirk of Circolize, and we're just handling it there. Here, like I said, this isn't super applicable to this particular analysis. It will be in my next video where I'm looking at Dragon structural variant calling, but this is just assigning a new column called color, depending on if it's a deletion, insertion, or break-end. They're all going to be black in this case because they're all break-ends, but this will work if you have different structural variant types. And then lastly, since this Circolize plot is only looking at chromosome 1 through 22 and plus X plus Y, we don't really want these contigs. The GRCH reference genome has a lot of different contigs that aren't associated with the whole chromosomes as we think about them. So I just filter those out for simplicity's sake. They may actually be biologically relevant, but for the case of just looking at structural variants across major chromosomes, we just get rid of these. And so next, here we're just kind of defining the different chromosomes for Circolize to read in. Circolize is actually not built for genomic data. This is a tool that you can use to plot a lot of different things, so we kind of have to hijack it to work with genomic data. And so we are providing each chromosome and then providing how long each one is, just to let you, so Circolize knows how long to make each of these lines. So as you can see here, they each get shorter, and that is because I am providing a decreasing length for each chromosome, which I pulled from the GRCH reference. And so then once we've done all this, it's time to actually start plotting. And so here I'm using a kind of weird way to extract this, but I'm starting a PNG device to save the plot. This is just a technique in R that I use to save figures. And then here I'm really just setting the parameters for Circolize in terms of spacing, height. This is pretty much everything I've optimized just to make it look good. When you do this without optimizing it, it kind of looks funky, and sometimes the chromosome names can not be fitting. So this just makes sure all the chromosomes are readable and where they need to be. And similarly, this is just affecting the font size of the particular chromosome names. So that's really, and I'm making sure they're on the outside of the circle, not the inside, since the inside is where all the important data is. And so then really this is the meat and potatoes of the Circolize plot here. We're just feeding it in this data frame, sans this line. We filtered that out, but I won't run that. We're just essentially drawing a line from this position to this position on chromosome 3. And we're just doing that for all of these. And these are going to be mates, so only one line will be drawn for each mate. And then here is, oh, also I'm denoting to color by break-in type. Like I said, they're all black here, but this will work with other structural variant colors. And then here I'm just creating that legend that you saw in the top right corner here. It will not automatically appear. And then here I'm just giving a quick little title. You don't need this, but I put it in there. And then here I'm just closing the PNG device, and that will essentially save it in your working directory. Wherever this RMD file is where the particular PNG output is going to be saved. So I'm going to just run this whole code chunk. You can see it do its thing. I swear. It will work for multiple different structural variant colors, not just SvABA. Alright, and so you know what to run. When it says null device 1, that means it saved the PNG. And so we can go look. Sorry, excuse my filming. But yeah, you can see 518. We just saved this. We can open it up. And there we go. That is the output from the SvABA structural variant caller. Please leave any questions if you have any. I think I've covered everything you need to know. I'm happy to help you all get this up and running if you need. But yeah, that's pretty much it for me today, and I will see you next time when I look at Dragon structural variant calling, and hopefully I'll do some compare and contrast between the two, see which one's better, and maybe look at some other ones. I've only used Dragon and SvABA, but let's look and see if we can learn some more. All right, well, that's it for today. Take care, and please like, subscribe, comment, tell your friends. I want everyone to learn how to do this. I think genomics is super cool. All right, but bye. I will leave you with that.